Good evening, everyone, and welcome to Immortality in Adelaide, the fifth and last of this series on the science of life, death, sex, and everything in between that's been presented by the Academy of Science across the nation. We've had an amazing time, but I have to say, this is the best looking audience we have come across anywhere in our fair country. That's right, Adelaide, you are rocking it here tonight. My name's Bernie Hobbs, and I am rapt to be your host this evening. We've got a terrific panel to help us delve into and tear apart the idea of immortality. What's in it for us? Is it a real thing? Is it ever going to be possible? What would it be like? What are the roadblocks? Is it something we want? Before we get there, we'll be starting in just a few minutes. I've just got a few thank yous to make. So I'd obviously like to pay my respects to the traditional owners of the land that we're meeting on this evening and to their elders past and present. And I'd like to thank RIOs who've given us this fantastic, and I don't know if you're all this beautiful in real life or if it's just the lighting here at the RIOs at the Science Exchange, but thank you very much, RIOs, for everything you're doing tonight. They're actually filming tonight's event as well, so everything will be up on the YouTube channel. We're also recording for Radio National's Big Ideas, so each of the 5 shows have played on RN, and you can hear them, you can podcast them anytime if you want to catch up on death, sex, murder, and something else, probably a bit too R-rated for tonight's audience, but no, not at all. Our partners you've already seen mentioned there. Our education partner is Nova, Science for Curious Minds, and our production partner, Science and Technology Australia. And the whole series has been put together by the team at the Academy of Science and led by one of our panellists this evening in a not very esteemed position at the moment, Professor Jenny Grays from the Academy, and also Professor Brian Fry, who's, I'm sure, much more decorous up in Brisbane at the moment. Immortality, it's a great note to end on. It's an intriguing idea, and not just for us, it's been intriguing humanity throughout the ages, and it seems appropriate here in the City of Churches to be looking at immortality. There's probably not a religion, I have to say, Wikipedia didn't look at all of them, but there's probably not a religion that doesn't have its basis somewhere the immortal soul, and certainly that soul featured heavily in the promises and threats of my Catholic girlhood. But we're not delving into religion. We're not looking at souls tonight. We're looking in a much more corporeal way at what immortality could mean for human beings and what it could entail and what those limits could be. The kinds of things we'll be looking at are what can we do to halt the process of ageing and permanently extend the life of a living thing? Can we revive long dead people? What about resurrecting extinct species? And just how close are we to achieving immortality? So, we've had the alchemists in the past looking for the elixir of life. We've had the Spanish explorer who went on a quest to try and find the fountain of youth, but accidentally discovered Florida, which it has been pointed out it's kind of the opposite of that. But, all praise to him for giving it a red-hot go. The search for eternal youth it's a bit of an obsession in our culture nowadays, and it's everything from exercising to make sure your telomeres can maintain their length to some wackier things like slowing your breathing to draw out your apparently allotted 1 million breaths per lifetime. Haven't got the source for that one here, but it's on the internet, so it must be real. As far as we know, there's only one species that's nailed, that's completely mastered the immortality trick, and that's, forgive me for the pronunciation, Turritopsis dohrnii, or the immortal jellyfish. But it doesn't just grow to adulthood and then stay in a lovely 25-year-old sort of version of itself for the rest of time without suffering illness or age. It gets old and then it reverses the ageing process and starts all over again. So it kind of sucks back its tentacles and its body shrinks and it literally goes back to not long after birth and then goes through puberty, once more. So I'm not sure if in the brochures for immortality anyone's really thought about whether that would be a price they'd be willing to pay, you know, not just staying at the life you've achieved now but having to go through the hell of those teen years once again. But that is the only species we know of that's been able to do it. We're hoping that science can give us some options that might let us skip that arduous process. Now, the question, science has in recent times given us some options that will help reduce our ageing, so reducing the age of immune cells and medications to increase bone density. Artificial organs we'll hear a bit about tonight, but is it possible to extend that extension of life indefinitely? Some of us may want it, some of us may balk at the idea of it, but is it possible? Our three speakers tonight are each working loosely in the immortality industry. It's certainly not what they'd be putting on their CVs, mostly, I guess. And they kind of represent three well-known from popular culture versions of immortality. You'll be hearing from science's answer to the builders of the bionic man. You'll be hearing someone who's a proponent of what I call the Walt Disney solution. And we'll also be talking a bit about the whole Jurassic Park idea as well. So we're hearing about techniques to lengthen life right now and into the future, and the challenges of preserving corpses to wait for some future breakthrough in reanimation, and lots of other stuff, looking in particular at capturing DNA to see if we can save extinct things or bring them back later. So that's enough from me. The way we're going to run tonight is we'll hear from each of our three speakers who will give you a very beautifully presented and engaging 10-minute, no pressure, 10-minute discussion or 10-minute talk about their take on immortality. And then we'll have a discussion up here for a while, and then we'll throw open to you. So please have your questions ready. Before we do get started, there are a couple more thank yous I forgot to mention earlier. One is to one of the brand newest friends of the Academy of Science, and that's Robert Jeffries here, who's one of our upcoming young scientists. That's right, wave, Robert. His sister Alice and his brother Nicholas and the whole Jeffries family. And Robert is a brand new friend of the Academy. He wrote and asked for a special present for his dad for Christmas, which is a pretty nice thing to do. And I encourage everyone to do that because they give this stuff away. So, and then you get a shout out at their next event. And behind them, not that he's written to the Academy, but he's a mate of mine, my not very godson, Chester Willis. How are you, Chesty? All right, nice to be in Adelaide. Now back to the serious business. Our first speaker is the guy you want to see if you've done some serious damage to your bones. He's well on his way to making the $6 million man a bit of a reality, although the budget has changed since the 70s. Professor Peter Chung is an orthopaedic surgeon and the director of orthopaedics at St Vincent's Hospital in Melbourne. Normally I'd probably stop there, but he does cop it from the Dean of the University of Melbourne if every bit isn't read out and every institution isn't named. So we're going to read a very long intro. Director of Orthopaedics at St Vincent's in Melbourne, head of the University of Melbourne Department of Surgery at St Vincent's Hospital. He's also chair of the Bone and Soft Tissue Tumour Service at Peter MacCallum Cancer Centre, and he's a researcher who's been involved in some incredible work which has major implications for extending life. I'll let him tell you about it. So please join me in welcoming from Melbourne for Immortality here in Adelaide, Professor Peter Chung.
Yeah. Thank you very much, Bernie, and good evening, everybody. My 10-minute starts with a recognition that mankind has been fascinated with the whole concept of immortality, and indeed the quest for immortality has driven the mystical and the medical. So, as a clinician, my perspective of life takes two forms. First of all, the simple fact of being alive, not dead. And that requires certain organs, your heart, lungs, kidneys, livers, and your brain, without which survival would not be possible. Then, of course, there's the second aspect, which is living. And that's my daughter up there, living and having a wonderful time expressing herself, connecting the senses of sight, sound, touch with movement so that she can interact with other humans as well as her environment. And it's when the failure of these systems occur that we look towards that question of how do we prolong life, how do we prolong activity. So the current solutions of staying alive longer, particularly if those vital organs are affected, your heart, your lungs, kidneys or liver, is really a mechanical solution. We have dialysis for our kidneys and liver. We have bypass for our heart and our lungs. And if those mechanical solutions fail or are not amenable for use, we look towards organ transplantation. And that's well established in the cascade of how we might deal with those systems that keep us all alive. Unfortunately, the brain has remained an absolute challenge. But because transplantation itself is a finite answer with its own problems, clinicians and researchers have really struck out on a visionary expedition to make a new organ. And so to make a new organ, tissue engineering occurs with 4 steps. The first step is to get a scaffold, the joists in a house, for example, to fill them with the appropriate cells, the brickwork or the cladding. Then to influence all that by exposing it to growth factors, I guess you call that the interior decorator. And finally, to stimulate that structure with the appropriate environment that compels those cells to go down the path of becoming what we intend them to be. And so the types of cells that we might use are the stem cells. And these are tiny little cells tucked away in some niche, waiting to be called upon to regenerate something or to create something. And a lot of studies have occurred and research to look at how we might regenerate kidneys, the liver, the heart, bones, and even nerve tissue. The explosion of technology around 3D printing, and we've heard all about 3D printing, which is actually based on a bubble jet printer, an inkjet printer. It squirts out a bit of ink, the ink hardens, it squirts out another drop on top of that, and so builds up a pile of layers of ink. And by printing such structures, you can actually create 3-dimensional forms like your ear, the shape of a kidney, a finger, for example. And more recently, there's been the introduction of cells into that ink. So you not only print the scaffold, you print the cells together with it, placing the cells specifically where you want them in that structure. So here's a lovely example of some recent research being done in the United States, where someone has taken a heart, an animal heart, dipped it into some solutions. These solutions have dissolved all the cells away to leave behind the scaffold that keeps its shape and supports it when it was alive. Then to introduce into that structure stem cells, which then populate the scaffold left behind, the collagen, and over time develop the ability to beat. So the development of the beating heart, the very essence of our survival, is now possible. And so researchers are really striving of how do we make this actually work? We've got the beating heart, and so that's where science is now going down. Let's take a look at function, the act of living, as it were. Bones, my particular area of specialty, make a very good example of how the movement of tissue from one part of the body, bone grafts, and bone grafts in the form of chips or bone with blood vessels attached, can be used to reconstruct the damage and the defects within bone. If that doesn't work, or if you run out of bone graft, or if you decide not to steal from Peter to pay Paul, you can actually put into these defects metallic prostheses like you see here in the arm bone. But in the world of personalized medicine, such as in treating this patient with a tumor in their lower limb, can you gather information, put it into a computer, the computer then accurately generates where is the tumor and where are the margins that we need to carefully resect around, put that information into a robot, which the surgeon then uses to accurately resect just that piece of bone that you need to remove. We then take that piece of bone and scan it, just like we scanned the leg initially. And using that information, input it into the computer, and that computer then generates the type of information we need to create a very specific prosthesis that absolutely matches the defect that we've created. Putting the information into a special 3D printer, and then using various systems of this, create the prosthesis that we need to fit directly back into the defect that we create. The so-called just-in-time prosthesis occurring in the next room while we're doing surgery, and this is a reality. Here are some examples of the type of prostheses that we've created and used in our patients, in the arm bone, in the shoulder blade, and in the heel, to replace bones that we've had to remove. Here's another example of how we can use 3D printing, a special 3D printer that a surgeon holds in their hands that squirts out a scaffold full of cells that will become cartilage to fill defects in joints that may ultimately produce arthritis, but to prevent it from doing so because we've repaired that defect. But everyone has their Everest, and mine is rebuilding limbs. I'm a surgeon who removes limbs from people from time to time because of cancer, and for that, there is an immense drive to rebuild that. What we have at the moment are very rudimentary prostheses that have been used and continue to be used for over the last 100 years. We also have robots that we can use and fix to people. But the challenge here is, can you get a device that hooks up with nerves? Can we 3D print skeletal muscle, the scaffold, populate it with stem cells, use 3D printed channels that come down that guide nerves into that muscle so that our nerves can control that specially tissue-engineered tissue, to look at how receptors can be created so that when you think about what you want to do, the tissue-engineered skeletal muscle then activates, sending messages to a robotic limb that might be attached to you. And by the same token, the movement of that limb and the touching of the fingers then send back the appropriate sensation that you read as the person who has that limb. So what does the future hold? Well, Australia is one of the only places that has both the bionic ear and the bionic eye as developmental products of our scientists. We have stem cells that we can inject in and help repair joints. We can tissue engineer organs and are clearly going down the pathway of creating new organs, tissue engineering nerves, artificial tendons, and now looking at muscle. We can 3D print bone in any type of tissue or material that you choose. So for the pharaohs of 1,000 years ago and their beliefs in immortality, it's taken thousands of years to unravel the mystery of immortality. For the medical people like myself, perhaps not so much immortality, but the prolongation of life and its activities is not only a possibility, but soon a reality in terms of what we can achieve. And I say to you, without even the introduction of this high level of science, how far have we come? Over 100 years ago, the median survival was 50. It's now 85. We have almost 50% increase in survival just by living healthier lives, by doing what we need to do to become a healthy society. In fact, both Australia and Iceland are the healthiest populations in the world. And there is a real sense that by 2050, being 100 will no longer be a stretch of the imagination. Thank you.
Thanks very much, Peter. Thanks very much, Peter. And that's not going crazily future. That's looking at what we can already do now and what we will be able to do in the very short future or very near future. And I now would like to invite, it was very rude of me to leave you standing over there, our other two speakers, Matt Fisher and Jenny Graves. No one should have to stand at cocktail hour, especially not when you're looking that glamorous. Jenny, if you could just have your seat there. And Matt's going to be our next speaker. Matt's not an orthopaedic surgeon, and I think his ears probably pricked up at the hearing of Iceland, though, because he is a big fan of cold places, hailing from Canada originally and over to Adelaide. Now he's in Melbourne. But Matthew is so dedicated to the quest of not just preserving life, improving life, but the quest for preserving life throughout perpetuity, even through death. And he's so committed to this that he's part of a team that's actually building a facility to cryonically preserve people after they die. Matt Fish is a software engineer who studied mechatronic engineering and computer science, and he's also the secretary of Southern Cryonics, promoting awareness of the cryonic preservation of the human body. Please welcome Matt Fisher now to talk about his vision of immortality.
Ladies and gentlemen, we are facing a global plague. There we go. We're all infected. There's no known cure, and so far, there's a 100% mortality rate. I'm talking about aging. Now, aging and death have always been with us. There's never been the prospect of a cure or even a decent treatment. And so we have no choice when we lose an elder but to tell ourselves stories to make it feel better. It was her time. She lived a good life. We mourn their loss and we move on. But people don't deserve to die just because they were born before you were. Human life is precious. It doesn't become less so over time. And yet, around the world, every day, 100,000 people die of age-related causes. That's 100,000 funerals, 100,000 families who have lost a loved one, 100,000 unique and irreplaceable individuals who are lost to the world forever. And to make sure they're gone, we burn them to ashes. Because that's what we've always done. It's all we could do. But what if we could do more? The history of medicine is the history of us finding ways to stop things killing us. 100 and some years ago, everyone died of the flu, tuberculosis, and diarrhea. We didn't know what a germ was until 1870, and they were killing us in huge numbers until we figured out how to bring them under control. And we did. Now we have to worry about cardiovascular disease, Alzheimer's, stroke, and cancer. These are what we call age-related causes. Wait, why is that jumping ahead? Oh dear.
That's okay. Take your time, we've got
Can
forever.
we bring that back?
Is it easier to fix it from there? Or?
We'll fix it from back here.
Thank you.
Thanks.
There we go. So in the last 100 years, we've conquered a long list of diseases like tetanus and polio and malaria. Howard Florey, born right here in Adelaide, gave us penicillin and saved 200 million lives. We invented X-rays and ultrasound, CPR, dialysis, and organ transplants. And now we're designing medicines based on individuals' DNA and 3D printing new limbs. Finding cures for the things that are killing us today is not a matter of if, it's a matter of when. And once we do that, we'll turn our attention to that fatal buildup of wear and tear that we call dying of old age. When that happens, everyone still alive will see their life expectancy start to increase by more than 1 year every year. We'll be able to keep people healthy indefinitely, which means we'll be functionally immortal. And there are people here tonight who are going to see that world. People who are young enough or lucky enough to survive the 50 or 100 years that it'll take for anti-aging treatments to do what it says on the box. It's going to be a world where you can't tell what someone looks, how old someone is just by looking at them. Where children don't find out about death when grandpa dies. They find out about it when we teach them about it in history class. It's a world of endless possibility. But some of us aren't that lucky. Some of us won't survive that long and will miss out on the chance to live a healthy life many times longer than any human has before. Now, I think that's a tragedy, and I want to do something about it. The only way to bridge that gap today is with cryonics, which at its most basic is using cold temperatures to prevent further damage to people who medical science has nothing left to offer. It's freezing people now so we can heal them later. This is actually the 50th anniversary of the first person to be cryonically preserved. There's 3 organizations around the world that provide the service, 2 of them in the US, 1 in Russia. And there's at least another 3 in various stages of development, one of which I'm proud to volunteer for here in Australia. And we hope to open our doors within the next 12 months. But there's a catch, as there always is. We can't prove that cryonics works. We can preserve people now, but the technology to revive them and heal them doesn't exist yet. And it may not exist for decades. And because we can't prove it, cryonics is not an alternative to a heart transplant today. It's an alternative to burial. You have to be dead to be frozen in the hope to be healed. Now, this might make it sound pretty far-fetched in the domain of eccentric millionaires, but it turns out cryonics patients are from all walks of life. They're a wide cross-section of people, just normal people like you and me, who have decided this is something that they want to do. Kim was 23 when she died of a brain tumor in 2013. Heinz was only 2. And Brian was 73 when he died in 2012. He was my father. "350 people like these have chosen to be preserved when they die. And there are another 10 times that who have signed up for when their time comes. The only thing they have in common is that they love life. They want a chance to keep on living. And that is the same reason that we go to hospital, the same reason that we take medicine, the same reason that we keep fit and we exercise, because we just want to keep living. Because when it comes to the crunch, if you've tried everything else, then you have a choice between 3 alternatives. Do you want to be burned, buried, or frozen? And only one of those gives you any chance of recovery. Now, hopefully, we couldn't heal Heinz, Kim, or Brian, much as we would have liked to and as hard as we tried. But now they're waiting patiently for us to figure out how to heal them, along with 300 and some other people. And hopefully, one day in the future, all of them will wake up with their family at their bedside, holding their hand, waiting and eager to guide them into the new world. Now, it could be a scary place. There'll be new technologies, there'll be different culture, there'll be unfamiliar people. But people adapt. The world is slowly getting more and more civilized as we learn to live with each other and learn to help each other. So I have hope. Cryonics isn't a cure. It's not even a treatment. It's an ambulance to take you somewhere that's equipped to help you, that just happens to be in the future. And once you realize that the deaths of everyone you care about are potentially preventable, or at least reversible, the scale of the problem really starts to become clear. The world's always been the way it is now, but it won't be this way forever. There will be a transition sometime in the future to a world where we take immortality for granted. Now, I want to see the other side of that transition. I want to live in that world. And I want everyone else to have the chance to join me there. Thank you.
Thanks, Matt. Thanks very much, Matt. And Matt, as you can hear, is not a crackpot. He's someone who has clear views and thoughts and reasons for his belief and his passion for working towards seeing immortality. I'm not saying that our other presenters are crackpots. Hopefully no one on stage right now is in fact a crackpot. I can guarantee you that our final speaker this evening, Professor Jenny Graves, is certainly far from crackpot status. She's a fellow of the Australian Academy of Science and, as I said earlier, one of the co-conveners of this series. And she is, even though she just I think received the mid-career award. She's been around long enough to have garnered quite a few subspecialties and eclectic areas of interest. So she was here as part of our sex talk in Melbourne. But tonight we've asked her to switch hats and take on or speak from her role as a member of the steering committee of Genome 10K, a project to sequence the genome of at least one individual from every single species of vertebrate creatures on the planet. That's about 66,000 genomes. She'll be getting some help with the project. It aims to understand vertebrate evolution and save dying species and preserve them for the immortal future. Would you join me in welcoming our third speaker, Professor Jenny Graves?
Well, thanks, Bernie. Well, I want to talk about the molecule, the component of us that really is immortal, and that's DNA. DNA has been copying itself for 3.5 billion years. So that's as close as we can get to immortal, really. We share this DNA with every other species on the planet, so that is 66,000 vertebrates, plus animals, plants, and bacteria. But of course, the sequence, which is what makes us us, has changed in all of these lineages, and it makes us a particular species. So DNA, we know a lot about DNA these days. We have a lot of DNA technology. DNA, we can purify. This DNA here was purified by a primary school student from mushed up peas, but you can easily prepare DNA from your own blood cells, and it looks kind of like white snot. So that's the DNA that we know and love. We know that DNA sequence is what makes us us. We know that it controls everything that we are. But it is just a molecule. It's just a chemical. DNA by itself can't do anything. DNA can't copy itself. It needs a whole lot of different proteins, even to just make a copy of itself. It needs more proteins to keep it safe. It's sort of like a house full of proteins that keep it safe and repair it when it breaks. To tell it genes that are part of the DNA, when to turn on, when to turn off, in the right place and at the right time. So you have this great big complex of DNA plus hundreds of thousands of different proteins all coming and going and being modified. And if that wasn't enough, and this is what we call a chromosome, because it's such a big body, we can actually see it down the microscope. But chromosomes are actually parts of cells. And if the chromosome is a house, well, a cell is a city. It has its own power plant. It's got walls and gates and channels and heavy industry and restaurants and garbage dumps. And the cell is a very complicated entity. But it's the cell that DNA needs to carry it forward from one cell to a whole body to the next generation. So without a cell, we really can't do anything with DNA. DNA by itself really can't do anything. So, too much. So cells, the main purpose of them is to transmit that DNA, that information from cells to other cells, to a whole body, to a whole species. And the way it does that is the DNA copies itself, and then the chromosome duplicates itself, and then proteins pull the two parts of the chromosome to different ends of the cell, and the cell then becomes two, as you see on this beautiful, isn't that gorgeous? Yeah, you see that happening twice here. And that's what builds our body when we grow, and that's what repairs our body when bits of it get old and die. So one needs cells. Again, we have a lot of cell technology. So we have DNA technology, we can sequence genomes, and we sequence the human genome and the mouse genome and the dogs and cats and kangaroos and gum trees, bacteria. We know a lot about the genome. We can also manipulate DNA. We can cut bits out, we can put new genes in, which is giving us a lot of new possibilities for medicine and for agriculture. And we can even edit little bits of the DNA. But we also have cell technologies. We can grow cells in flasks, in an incubator that keeps them warm. We can manipulate cells. We can stain cells different colours. For instance, the DNA here is stained red, so you can see the chromosomes. And we can observe how cells act. Well, cells come in many different sizes and shapes, and the cells of the body have many different functions. So the cells in your brain are those that have electric currents and help you think, but there's cells in your muscles that are contractile and help you move. There's cells in your liver that make handy proteins and put them in the blood supply. So cells in the body generally will differentiate and take on particular roles. Well, once a cell is differentiated, it can't change its mind. Neurons don't become muscle cells, and they can't go backwards to being an embryonic cell that can essentially do everything. So differentiated cells in the body, that is it. When that body dies, they die too. So differentiated cells, which is most of your body, that's what we call somatic cells, they will die with the body. And the only cells that will actually carry your DNA into the next generation are what we call germ cells. That's the egg and the sperm. And the egg is a very, very large and complicated cell loaded up with nutrients. The sperm is basically just a package of DNA with an outboard motor that delivers it to the egg. And then one sperm fertilises one egg and produces the embryo. Again, we have technologies now that we can look at eggs and sperm and embryos in a culture dish. We can even manipulate them. We can change the way they differentiate. We can observe the way they differentiate. We can even swap the nucleus of the egg cell for another nucleus. And this is something called transplantation that has been worked on for many, many years. You can actually put a nucleus from one of these differentiated cells that's going to die. You can take that nucleus, put it in an egg cell, and trick it into thinking it's reborn. And it can then form a complete animal. And I'm sure you've all heard of Dolly. Dolly, the sheep, was a clone isolated from a single cell from a mammary gland and implanted into an egg. So we have all these awesome technologies with cells. But most cells are going to die. They're going to be burned or buried or maybe frozen. And it's only the germ cells that normally carry you into the next generation. But there's an exception, and that exception was mentioned by Peter, and that's stem cells. We know that the embryo is full of stem cells, that these are cells that haven't decided yet what they're going to be when they grow up, and they can turn into anything. And it turns out there's actually little packages of stem cells in some of the organs as well. And even better, it turns out that you can actually trick an ordinary skin cell, say, into thinking that it's a stem cell by giving it a particular cocktail of genes and proteins. So we have these induced stem cells as well as these embryonic stem cells. And again, that's going to make a huge difference in, for instance, repopulating some of the 3D frameworks that you've been hearing about. But also these stem cells, you can trick them into thinking that they're a part of an embryo. So if you squirt one into an embryo, it says, okay, well, I can be a part of this mouse or maybe this human, and they can go ahead and differentiate into a wide variety of cells, including even germ cells. So it is actually possible to take cells, and for instance, you could take cells from my skin and grow them in a culture dish, and then you could add this cocktail and turn them into stem cells, which are these little orange clumps of cells. And these cells then think that they can do anything, and you could in fact implant them in an embryo and clone me. So I just want to say that
You are not possible.
that is this immortality? Well, no. My clone is not me. I will still get old and die, and my clone will go soldiering on, and my clone might look like me, have blue eyes and long, long hair, but my clone is not me. And in fact, there's many, many things that happen to the genome that actually change the genome. So this clone will have a different mother. The mother will have a different diet. There'll be different insults that happen to this child's DNA. And so growing up, that child will not be even genetically identical and certainly not physically identical to me. So we can do all of these awesome things, but they don't really amount to immortality as on an individual basis. Can we look more generally at immortality of us as a species? Can we look more generally at species that are endangered? Can we rescue them? Can we bring extinct species back to life? And of course, it's always been my vision of lovely to go to the zoo and see a Tyrannosaurus Rex, wouldn't it? So, in fact, we have lots of awesome animals that have become extinct. Some of them 100 million years ago, like the dinosaurs, some of them more recently. And you'd have to say, well, there's really not much left of dinosaurs. There's no DNA. There's fragments of protein, but largely it's just fossils, rock-like bits and pieces. So we really can't see a way of generating a new dinosaur. But it might be different with Ice Age mammals. For instance, there are mammoths, mammoth calves that froze themselves by dying in the tundra and have been deep frozen for 10,000 years. And in fact, there is DNA there. And in fact, the whole genome of a mammoth has recently been put together and published. So we have a genome. Do we have a mammoth? Well, no. Unfortunately, we don't have any chromosomes, we don't have any cells, we don't have any eggs or sperm, and certainly no embryos. So we really don't have the wherewithal to make a mammoth. We can tell a lot about what a mammoth was like from its genes, and we could even take a little bit of that genome, a gene or two, and put it, say, into an elephant stem cell. And maybe we could make woolly elephants, but they would not be a woolly mammoth unless we took every one of those 20,000 genes and put 20,000 mammoth genes into an elephant stem cell, which is really a little bit beyond what we can do at the moment. Well, what about species that became extinct quite recently, like the thylacine? The Tasmanian tiger has only been extinct for 80 million years, and there's tons of tiger stuff all over Australia. Every museum's got dozens of tiger skins, there's pickled embryos in bottles. Can we use that material to bring back the Tasmanian tiger? Well, again, we've got tons of DNA. The thylacine genome has been sequenced. It's about to be released. It's quite a good sequence. So we're going to be able to tell quite a lot about what tigers were like and how they behaved just from their sequence. But again, no chromosomes, no cells, no embryos. So it's not going to be possible to recreate a thylacine. Again, we can take thylacine genes and maybe we can put them into a Tasmanian devil and then see how those genes react. But again, to make a tiger, we'd have to have 20,000 of them. And again, that's really not on at present. Well, so de-extinction is still a bit of a myth, but would it be possible to take endangered species, the ones that we haven't quite driven to extinction yet, give us time, we can get their DNA, we can get their sequences, and we've done that with the giant panda, we've done that with the Tasmanian devil, we have the whole genome sequence, but we can also take cells and grow them in culture and freeze them down, and it's easy to freeze down cells. I've done that thousands and thousands of times. Single cells freeze down very easily in antifreeze, and you get pretty good recolonisation when you thaw them. So can we do that with endangered species? Well, yes, people are doing that. And there are many establishments all over the world called frozen zoos. This is a big one in San Diego Zoo, in which they have many, many, many DNA samples, cell samples, egg, sperm, and even embryo samples of species which are endangered. And Genome 10K is certainly trying to help by getting the genome sequence of all these endangered animals. But frozen zoos are really not much fun. I mean, you're really not going to get the crowds bringing their kiddies to see a whole bunch of liquid nitrogen cylinders, and even if you look in the cylinder, you see a whole bunch of tubes. So it's quite expensive and difficult to keep these going. And of course, if there's a power failure or somebody forgets to top up the liquid nitrogen, that species is extinct forever. So we have to think about, well, what can we do and what is sensible to do? What is it very expensive to do? Are there better ways to save species? So in conclusion, I just want to remind you that we know lots about DNA, we have lots of sequences, but we can't make a chromosome and we can't make a cell yet. So we have to have ways of putting DNA into cells and embryos that are pre-existing. And we have to think about where this technology is going to take us, what is worth doing. We have to think about extincting species and what we would do to save them. We might even think about humans as possibly an endangered species, and maybe we should think about freezing down human samples in a frozen zoo, so maybe some friendly gorilla would come and bring us back to life when we stop killing each other and ruining the planet. Thank you.
Thanks, Jenny. Painting a beautiful and quite detailed picture there of the past, the present and the potential future, Professor Jenny Graves. And I like how you tied in the ideas of the freezing and the liquid nitrogen and the power failure as well, which is something that Matt's spent at least a little bit of time thinking about and planning for. Now, what we'd like to do now is have a bit of a discussion amongst ourselves, just to bring out some of the detail from what you've said and then throw open to you. And I have said we won't get bogged down in philosophy, but I really do want to put one question. It's such a big topic for us. Can I just ask in the room here, who would like to be immortal? Who would like to live forever? Okay, so... we've got every one of the panellists, Jenny with the proviso that if she could top herself if she got bored. And can I just see the hands in the audience again? Maybe about 10%, I think. And you'll be happy to know there's a gender mix as well. So it'll be a fairly interesting place when the big thaw happens. Now, I'm not someone who does want to be immortal, but I wonder if there's a version of immortality or a version of extending life that appeals to you. You've said you go for immortality. What's the version of immortality you'd go for, Peter?
Well, I love every day that I'm alive. I love doing everything I do, and in fact can't wait to fill every single moment. Drives my wife crazy. drives my friends and colleagues crazy. And I often say to them that it's that quest just to keep doing more and the interest that we get back from it that inspires me to keep going. And I often think about at what point is enough enough. And I would suspect it's when you're really tired. And when you've
Have you ever been tired?
Not of what I do, and not of the interest. And I think that's the thing that really, really drives you. And so for me, life is so full. I guess if you switch the switch off today, I would have had a complete life. But the fact is, if the life going forward is as good as the life I've just come from, I'd just love to keep going.
Keep going. So you wouldn't put a full stop on it
No,
as long as it keeps going?
I wouldn't. Having too much fun.
Matt, what's your answer to this mystery question that I can't imagine what you will say?
I agree completely with what Peter said. I'd like to keep living until I decide that I am done and I don't want to live anymore. At the moment, I can't imagine that day coming if I'm still fit and healthy and active. So basically, until I decide otherwise.
Okay, ball's in your court. And Jenny?
Well, I think fit and active sums it all up, because I think it depends very much what your quality of life is. And I think many people who, like me, have gone through health crises where you can imagine that you would not wish to live, would be very cognizant of drawing a line between what is really worthwhile life and what is really not worthwhile life.
Wow, well, I'm clearly on the outer because I really have no wish for it at all. I mean, I'm very happy just to have my 70, maybe a couple more. But I think if I was to go the ideal lifespan, the big problem I would find is that different parts of us peak at different times. So I think I'd want my knees from when I was 30 and maybe my emotional intelligence from when I'm about 75, because man, is that a journey. And I think my mental peak would be somewhere in the 40s. So I think it's a really tricky thing to find that bit of your life that you would want to keep going at forever. But anyway, I did just want to just get a sense of what your thinking was on the immortality thing before we dig down into the science. And
how about a less confronting question of, would you like to feel 10 years younger? And if you would like to feel 10 years younger now, do you think you'd like to feel 10 years younger again in 10 years' time?
Is that multiple freezings?
No, not freezing. Just instead of thinking, I don't want to live forever, just think, how would I feel if, someone offered me a pill that made me feel 10 years younger now?
Have you got that pill?
I don't.
Okay, just checking. Okay, so I think that is a much more interesting way of looking at it as well, and certainly might help me get around my knees and what have you. I hope you're going to be there in the future because I'll be needing those orthopaedic skills, I promise. So, Peter, you took us through quite clearly what we can do now, and you gave us a sense of what we can look at for preserving our lives and maybe helping these old bodies stay in shape for a bit longer. But the idea of replacement organs to order, the sorts of thing that you were talking about, how far are we from the day when I turn up at the hospital needing new kidney, new liver, whatever, and you can just make it happen?
I actually think the realist in me will say we have quite a way to go yet, simply because we have not understood the finer details of what makes organs work. We can rebuild tissues, and you can see from the science that's been published that the concept of organ manufacture is definitely there, and it is a reality. But to make it work in concert with the rest of the body in a way that makes sense and prolongs the efficiency and effectiveness of that function is what it challenges us. And I think the wonderful thing about science is that in the past, we couldn't even dream of these things. Now we can dream about these things, and technology is keeping up with us. And things that we thought were just simply impossible 2 or 3 years ago, we can do now. When we started 3D printing 5 years ago, 6 years ago, little things, we had these rudimentary 3D printers that really were the HP inkjets that were modified. And now we're actually 3D printing 3 different jets within each other at the same time, little packets of cells coming at different points. And the sophistication of technology, and I think we've reached the J-curve in technology, it is now taking off. And I guess that's what the excitement is.
So I guess the catch in what you're saying there is, yes, one organ at a time, but so say if I wanted to come in and get, just say we had the technology, right, I want you to replace all the bits that I've done the damage on right now. We don't know how that would work, if that would give me another 50 years of life.
No, we don't know, but what we do know is that cells have as we know them now, and the manner in which cells survive in the cell cycle, is that there is a timeline to cells. They senesce, they do grow old. And if we are to be immortal, every part of us has to be immortal, not just the replaced kidney. The skin has to be immortal, the eyes have to be, whatever it is, and it has to go along with you. You have to grow at the same rate, as it were, or be replaced in a way that allows you to continue to function at the same rate. Your heart can't outlive your body. It has to move at the same rate.
And I guess the key question is, if you keep replacing all those bits, when do you stop being you? And you've said that your Everest is the limb, but surely replacing a brain would be the real Everest.
Look, orthopaedic surgeons are
that's
constantly challenged about having a brain, so that perhaps is not so important for me, but my hands are so important. No, look, I guess the limb is my Everest simply because it's within my sphere of influence, but I am sure the brain, which is protected by the hardest piece of bone you can come across, for a very good reason. Yeah. It's for a very good reason. It's in the hardest part of your head, or your body. It's because it is possibly the most vital central node that commands everything else.
And it is difficult to imagine with every single neuron having upwards of 10,000 connections that all have to be right. But
can I say that the computer companies, IBM, so on and so forth, are developing chips that are so fast that they can make the connection. In fact, in order for them to have a chip that you can implant that does the function of the brain, they actually have to slow the chip right down, because it's the speed of the chip that releases a lot of energy and requires a lot of battery work. They actually have chips now that can work as fast as the brain with the same number of neural connections. So you can see how in the future we'll be implanting things to replace almost brain function.
Okay, I think we need a little reality check on the future science here. Peter, I didn't know you were... I thought you'd be grounded. Orthopods. Aren't you all about union, rugby union and, I don't know, nights out with the boys? Jenny, you've effectively put the kibosh on the great stories of bringing back extinct species from bits of DNA. I've never heard that put so clearly or beautifully, so thank you for that. Am I right in saying, from what you've said, that unless we have got viable tissues, viable cells from an organism, there's effectively nothing we can do to really bring that creature back. And even if we do, we're only bringing back a clone, an identical twin of it. We're never bringing back the original. So the best hope is for a species to come back from actual living tissue that's been frozen.
That's true at present, but of course what we all cell biologists would love to know is how do we build a chromosome. You know, we've got a metre of DNA chopped up into 23 bits. We know basically what proteins wind it up, and we have a pretty good idea of about 100 different proteins that glob on. Is there any way we can actually build it up from those components? Is there any way we can actually build up a cell? Maybe we can 3D print a cell scaffold. And of course, there'd be lots of reasons for wanting to do that, just to know how the cell works, just to know how the chromosome works. And if we could do that, making artificial chromosomes or artificial cells, then the picture would look very different.
So that's clearly the next step if you wanted to go along the thylacine.
It's a very big step.
It is a very big step, but we're here with big minds, big imaginations, here as part of the Adelaide Fringe Festival at the Science Exchange at RIOz. Now, Matt, you are clearly very serious about immortality through cryonics, and where are we? Well, I'd say most of us, obviously there are some people with real interest in it here, but for people like me, the most we've ever heard about it is, the Walt Disney factor that the guy had his head frozen. But it turns out that's not strictly true. What's the catch with Walt Disney and cryonics?
Well, he was looking into it in the last months of his life, but he had lung cancer and he had a lung removed, and he died quite a bit faster than the doctors thought he would. So he didn't manage to get the arrangements in place in time, and he was cremated.
So Disney, not frozen,
Afraid not.
burned, all right, and missed out by just a couple of weeks.
Yeah, like 2 weeks later, the first cryonics patient was preserved. His name is Dr. James Benford.
All right,
In
so
1967.
1967, so 50 years. 50 years ago, this year, we have the first cryonics patient, I guess, first cryonics entrepreneur or explorer playing their part. In those 50 years, can you tell me where are we at? What's the most advanced life form that has been, because you yourself say we're a hell of a long way from having the technology to bring something back from the frozen.
Yep.
What's the most advanced life form that has been frozen down and then reanimated?
Well, it's been done successfully with nematode worms, and they've proven that they retain the memories that they had before they were frozen, so that's very promising.
I love that they know what nematodes remember to
Yeah.
start with.
Well, they taught them to recognize a certain chemical, and they still recognize that chemical afterwards. It's also been done with a rabbit kidney. So they took a kidney out of a rabbit and froze it and thawed it and implanted it back into the rabbit and removed his other one, and the rabbit went on living. So it's been done with organs and microorganisms, but no mammals yet.
No.
We're all waiting for that day.
Okay, so we are waiting for that day. So I want you to talk me through what it is that you would go through. Obviously, you'd have to die first. Talk me through what your vision is of what's ahead when your time comes, the first time. And then I'll ask Jenny and Peter to give us a bit of a science appraisal of any blockages that they see in that plan.
Right. Well, ideally you'll have a team from a cryonics organisation waiting by your bedside in hospital, waiting for the doctor to declare legal death. And then they can go into action. They put the patient in an ice bath. They administer anti-clotting medications and wash out the blood and replace it with a cold saline solution to get the body cooling as quickly as they can.
Can I just interrupt? So far, so good. Everything sounds doable there. Yep. Okay, good. Please keep going, Peter.
The patient's transported to the cryonics facility, where they're perfused with what are called cryoprotectants, which is essentially a medical antifreeze. And the temperature is lowered in a controlled way down to minus 196 degrees Celsius, which is liquid nitrogen temperature, and that's over a number of days. And once they're that cold, then they're placed into liquid nitrogen in a vacuum flask, essentially. And they wait there until medical science advances.
Hopefully with a good backup generator and...
Well, it's not reliant on power. It's a passive bottle full of liquid nitrogen. So you do need to keep topping up the liquid nitrogen every few weeks, but there's no refrigeration system per se.
Okay, assuming the logistics company charged with maintaining the liquid nitrogen, let's just assume that that's not a problem at all. And then the hope is, how is this sounding so far with the liquid nitrogen freezing process?
In principle, that's what we do with cells in our labs.
That's what we do.
Yeah.
Thousands of times, but I have to say I've never managed to keep a cell line going for more than about 10 years before the power goes off or the technician forgets to top it up.
Yeah, so that's all right. Your group that's building the cryonics, they'll be very focused on making sure that there's very good technologists topping up the liquid nitrogen. All right, so let's fast forward to, give us a time, 500 years, when you think, is that a safe sort of bet?
I think there's about an even chance of it happening within 100 or so.
Okay, let's go split the difference, 250.
Yeah, okay.
Okay, so in 250 years, what's going to happen?
Well, hopefully by then the technology that you need to revive someone, these treatments will be much cheaper than they were when they were initially developed, as these things tend to do. And we'll be able to revive everyone who's in storage, starting with the people who are most recently preserved, because the technology to preserve them would be better than the technology that we're using now. So it's most likely going to be a first in, last out sort of process. And it's difficult to speculate about what technology is actually going to be required.
But what would you hope would be the outcome? You wake up as?
Right. That level of technology is probably going to be similar to the kind that you would need to rejuvenate the body comprehensively. So you'd be able to fix the skin and the bones and the organs and all the other things that slowly go wrong as we get older. Which means that hopefully you'd be waking up fitter and healthier than you've been since you were 25. And you might look like a 25-year-old if you chose that, or if you said, no, I want my face to stay the same, then that could probably be arranged as well. But
the default option, I think it's the cheaper one. Okay, sciences. Do we have any issues with, I mean, just apart from the fact that, as Matt said, none of these technologies are around yet, can you see any blockages that would, just roadblocks that just go, nah, that can't happen, that cannot happen? Or is it really a matter of seeing, can this be done?
There are so many unknowns, and there are lots of unknown unknowns, as was famously stated. I mean, we really don't have a very good handle on ageing. I think there's a great deal we need to know that we have no idea about right now. I don't see that as a blockage, but it could be a much longer process than we think.
I'm glad that no one's saying it'll happen in the next 5 years. So the standard medical research answer. Peter?
Well, I'm absolutely inspired by human capacity to achieve, particularly when they sit down and work it out. So the Human Genome Project, just the concept of that, when I was just a little boy, was science fiction. It never happened. How could you possibly do that? They thought it would take 2 generations. They did it in 3 years, I think. You know, 8 years. So the capacity to achieve and solve a problem is now being multiplied by the fact that we have technology to use to achieve those things. I think one of the things we have to reflect on, though, is the intangible. What about the psyche of the person being introduced into a new environment? I mean, what steps should we put in place? As we reanimate, as we rejuvenate, what do we do about the thinking process and the emotions and the cultural changes? If you can imagine now living in a society where people are immortal and you've come from a society where you're not, how does that change the way you might think about the value of life, the sanctity of life, what it means to have babies or not have babies? So those questions become very, very important. And I think it's not till you can't have something that you appreciate how important it is and how much you give up of the value when you can just pick one up at the shop down the corner. And it would be a very interesting perception of life if you could have it forever.
You so don't talk like an orthopaedic surgeon. This man's got soul, yeah. I'm interested to know now if you would, because you've said quite a lot about being, you know, loving life and all this sort of thing. Would you sign up for cryonics?
Well, living forever, conceptually, living forever means a continuing process of living, of picking up and changing. Being cryonically preserved after death is not living. It's just an opportunity to be reanimated if the technology arose. And I think it won't be within my time, but I really...
But if it was?
If it was, you know, one is really tempted to consider that. I'm a bit of a futurist. I was brought up in the good ship no can do. So when anyone tells me you can't do something, that is precisely what we're going to be trying to achieve over the next 5 years. So you know, I don't get put off when someone says it's too hard, don't know. Well, go out and find out.
Just so you know, Matt is sizing you up for the steering committee right now. He's so got your number.
He's measuring me up, I think, more.
That looked too Undertaker-y, yeah. Oh, right, yeah, the big thermos, gotcha. Look, I think there's only one thing that we haven't touched on yet, and that is it's a pretty dirty topic, but we do have to talk cost. I mean, in both of these areas, I mean, I think we can let go of the species thing for the time being, but looking at rebuilding a complete bionic, is that something that's only ever going to be affordable by the very rich?
No, not at all, because if you put it in context of where we are today, and let me give you an example. About 40 years ago, when they started to do joint replacement surgery, people used to wonder, how much is that going to cost? That's ridiculous. We couldn't possibly afford that. But every year in Australia, 100,000 people march up to get new joints. You know, it's capitalised around the world. $30 billion is being spent on that, and it's part of the budgets of life, let's call it. So, you know, what we're doing now is overly expensive because we're researching it. But there is no question for it to become viable into the future. There's a health economic facet that we have to consider. We don't drive F1 cars, but because of F1 racing, we drive safer cars.
Okay, Matt, do we have to have a Disney budget to be able to afford cryonics?
Well, the organisations that offer it at the moment charge between 30,000 US and 200,000 US, depending on where you go.
And
but
for how long?
in perpetuity. So the model is that if you make the payment when you need the service, then most of that goes into investments and interest-bearing accounts and pays for the ongoing storage and administration costs. We hope to be able to offer it for about $90,000 Australian, and most people pay for it with life insurance. So you get a life insurance policy for $100,000. It costs you a few hundred or $1,000, $2,000 a year, and then that goes to pay for your preservation costs when you die.
So as long as capitalism and liquid nitrogen are maintained, there's no flaw with the system so far that I can see.
None whatsoever.
No.
Guaranteed.
Look, it's over to you now for your questions. I'm sorry, we have gotten a bit carried away up here. We've got plenty of hands shooting up. I will ask you, no comments, thanks, just questions. We want to get as many as we can. Our microphones are coming. We've got a microphone up top as well, and we've got one lady right here, one lady there. Yep. Thank you.
Matt.
That's all right. If you keep talking, they'll turn it up.
Okay. Matt, there's two things. There's how to reanimate and also how to cure what you died of. If you've had invasive acute myeloid leukemia and none of your bone marrow is left, how can you suddenly reanimate that person and instantly cure what they died of?
Dr. Matt.
Well, obviously the technology doesn't exist yet, so it's hard to speculate exactly what it will look like, but probably a thorough rejuvenation process over days, weeks, months even, while you're still unconscious, doing whatever repair work needs to be done to the body, possibly with molecular nanotechnology, if we want to use that buzzword, but you wouldn't be woken up until we had the technology to do all the repairs that were needed. So you can just wait another 50 years if you have to.
You'll be looking great. So upstairs, if we have one upstairs, we may not be able to get the camera to you, I'm sorry.
Okay, can you hear me?
Yeah.
So, look, I guess, I know we weren't going to talk philosophy, but I guess one of the elephants in the room is population growth, and what, can we actually all afford to keep living when we keep, populating?
How does the maths work out? Because you were saying 100,000 people die from age-related illnesses each day, but there are also 350,000 people being born each day at the moment.
Well, the birth rate in developed countries has gone down a long way, and we're now below replacement levels in a lot of places, even in China. They're struggling to work out how to grow again. But people only don't die once, but if you have kids, then they can have kids who can have kids who can have kids, and you get much faster growth from a slightly higher birth rate than you do from lowering the death rate. So I don't think the problem will be as bad as we fear it might. And on the other hand, I would grant more right to live to someone who is already alive than someone who may someday be born.
I have a feeling evolution has a very good reason for killing off old people and letting new people coming through, something to do with resilience, adaptability, openness to ideas. I'm just saying, I have a feeling there would be something in there, but we will go to the next. Oh, Jenny, quickly. Yep.
Well, it seems to me that it's not just a numbers game, it's also a population stratification game. I mean, your 900-year-old Methuselahs, are they going
Let's call him Rupert Murdoch.
Are they going to be the bosses? Are they going to be the kings? Or are they going to kind of take a back seat and let a 200-year-old run the place?
Yes, I can't imagine what way that could possibly play out. Yes, we have a question here.
Yeah, following up that last comment that you made, would an immortal society be deadly dull?
What are your thoughts on that, Matt? Because you're the one who's most likely to be there.
Like, turnover of ideas and ongoing change is definitely one thing that death does bring to society at the moment. But if we imagine the society where people don't die and we have the problem of stagnation of ideas, I have to ask, would we come up with the solution of making people get old and die by the time they're 80 as the best way we could possibly solve that problem? Or could we find some other way to keep new ideas coming into society, like maybe put social pressure on people to retire or change careers every 50 years? I think there are better ways to solve the problem than executions.
What, the Logan's Run solution? Yeah. Peter.
Just a thought that one gets the sense that when you talk about immortality, we don't think about a growing life of bringing in experiences and changing with those experiences as we all do every day. There seems to be a sense that when we talk about immortality, people think you just keep growing older and therefore take on our stereotypical perception about what being old is. Now, I attended Sir David Attenborough's presentation last week, and I was wowed by the greatest naturalist, this living naturalist, 90 years old. He was taking in new ideas. He was talking about drones and camera angles and all those sorts of things, and it shows the active mind acquires information and is prepared to change with it. And I think in the concept of, will it be boring? Should we do it? Should we not do it because of population growth? I think science has to marry really well with the ethics of science and the ethics of humankind to come up with the right answer of what we do. We don't do it because we can, we do it because we should, and that really is the amalgam of culture, ethics, honesty, thinking beyond what science has to offer.
Here's another tip. I reckon you should see if you can sign up David Attenborough, because if you do that, you'll have a lot of other people interested in going along, namely your steering committee guy here. Question from young Chester.
Well, basically, when you come out of cryo-freezing, wouldn't all your old possessions, you'll basically be rebooting your life, because everything you've owned in the past would be either gone, old, outdated. You'd basically be stuck.
God, think of the updates list on your phone. It's just...
You'd have to have a lot of money.
Yeah.
It's true. You'll have to adjust to the new world, and you won't have brought a lot with you. There are ways you can put money away so that you have money when you are revived, and we can also organise to store some personal effects for people, so they might store, some favourite photos or a flash drive full of whatever they like.
A flash drive, yes. No, I'm just thinking what they'll think of it in the
What is this technology?
2500.
And my dog.
Yeah, well, we might take pets as well. I mean, in fact, we plan to take pets. But in terms of the experience that you'd go through, I liken it to that of a refugee, in that they find themselves, they can't stay where they are because they were going to die. They go through a dangerous journey to a new place, a new country where they may not speak the language, they're not familiar with all the technology around, they have to adapt to the culture, they may not have useful skills, they may not have family, they may not have friends. But people do that all the time because life is what it's all about. Life is the most important thing, and people will do a lot and go through a lot to keep living.
Well, I certainly hope you have more luck establishing that welcome place for people in Australia than we have with refugees
Me too.
lately. Time for one or two more questions, yeah?
Hi, just wondering how far away would we be from designing human embryos with genes that are more genetically superior to create a more healthier or fitter individual as to prolong life more?
Jenny?
We basically don't know what genes will do that, although we've begun to isolate a few that seem to prolong life in yeasts and mice. But probably environment is just about as important, and the environment we now know actually has an effect on the genome. So if your grandfather was starving, you're more likely to be obese, for instance. So there's lots of things we just don't know about that aren't actually genetic.
These are epigenetic
These are
effects that you're talking
epigenetic
about.
changes, and they're probably just as important to know about as what genes you would implant in the embryo. So I would guess one of these days we will have lists of genes that will prolong life. Probably that's mostly just fixing up very tiny little small problems that people have in their genes that restrict life. So if you can get a blank slate of those genetic variants that actually are shortening your life, you'll then presumably prolong it. And there is a lot of interest, of course, in fixing up genes which are desperately bad for life. And eventually, I guess we'll get to variants of genes that are just not good for life.
In the meantime, don't smoke. And also,
way more important.
half an aspirin a day is what the doctors take. So, we have another one here, yeah.
What do you think of the prospect of like digitally storing your brain to like get revived in the future with mechanically or biologically like storing your DNA, like regenerated in a time where you can actually clone your body and restore your mind from being stored online or in a computer somewhere for the future?
Was it more than a great book?
Well, uploading is certainly a possibility. It's really tricky to speculate about what technologies are going to develop first to the stages where they become useful in these kind of ways. But there's a lot of people thinking about how to extract the information from a brain and reinstantiate that in software, and whether you run that in some sort of virtual world or in a robotic body, or even copy the brain atom by atom and put it in a new cloned body, who knows? They're all possibilities for how this could play out, but they might be a bit further down the track than reviving someone in optimal conditions and continuing life in their own body.
That is great that we've found something that's less likely than being able to reanimate a frozen body. That's good. Yes.
I'm fascinated by clones. You mentioned that if you made a clone of yourself, you'd be raised by a different mother and have different things assaulting you during life, so it wouldn't be you. But you could get, say, 100 dollies, put them in the same pasture, bring them up in the same conditions. What differences would you see, if any? Well, like, I'm interested in exploring differences in clones, basically.
Probably some quite subtle effect, like where was the embryo in the uterus? How good was the blood supply? Things that are really difficult to control, and they have a big effect on the growth potential and the rate of growth and so forth. With Dolly the sheep, we probably have a lot more control than I would have over my clone, who would have a different mother, who would possibly smoke and eat the wrong things, and that would compromise the baby's existence.
Thanks, Jenny. We're going to go for a few really quick last questions. So,
when I was maybe 4 or 5 or 6, that first time I thought, hey, I'm going to die one day. My next immediate thought was, well, I want to live forever. So I can't understand. I'm struggling to understand the people that want to die. And if I just consider your case, let's say you get the new knees, you your heart starts to go, then your heart... So actually, modern science actually gives you the opportunity for immortality. So why would you arbitrarily kill yourself at 85 or whatever the expected life is?
Fair point. It's not, I guess, for me, it's just that whole cycle of life thing and being part of a much bigger picture. Like, it's not just all about me. Like, there's limited resources on the planet. And, oh yeah, I do get tired, unlike Peter. So it's about... and having lost a number of people I love as well, it's about seeing life as more than just my life and wanting, whatever role I play here on the planet. I think, it's of limited benefit to everyone and everything else on the planet. So I'm quite happy with being part of a cycle. I don't imagine there's anything I'm going to afterwards. I fully expect I won't be frozen. I fully expect to be in the dirt somewhere, and that will be it. Bye-bye, and it'll just be the effect I had on people that lives on, but that's just enough for me. So we just had different reactions when we were kids, I guess. There's very quick last one there, or second last one there, and then last one here. Thanks.
Just one for Matt. I was just, the Centre for Cryonics, I'm not really sure how it's funded or how it's set up right now, but if I was a potential customer and I wanted to put my money down, I'd want to be assured that this company was going to last, 500 years or 250. What would be the, have you thought about that? It's like, is it going to be aligned to governments, or is the government going to be looking after this, or
we can assume he has thought about it. Can you maybe tell us what you think will be the solution?
Yeah, absolutely. We thought very carefully about how to structure the constitution of the company to make it, give it the best chance of lasting as long as possible. So anyone who's on the board has to be actually signed up with the company, so there's no motive for people to come in and steer it in the wrong direction. So all of us involved are signed up to be preserved by the company, and that gives us a very strong incentive to make sure that it's as long-lasting and as reliable as we can. We call it building our own lifeboat. Also, it's a not-for-profit, so we wanted to make sure that there would be no corporate takeovers or anything like that. And it's a real challenge, though, and we need to be very conservative in what we do and what we try to make sure that the company just continues for as long as it needs to.
Terrific. And our last question.
I just wanted to, if I might, make two comments about cloning.
What did I say about comments?
I know you said no comments, and that's why I was asking, but if I could. And so...
I know. They're not my death eyes.
That's why my question was, can I make two comments?
Yeah. Okay, because it's you.
Thanks, Bernie. First comment, what Jenny said about cloning livestock is spot on. They have cloned a lot of dairy cows, and they are indeed sort of like identical twins, and with this fortuitous differences in epigenetic changes that you'd expect. The other thing I'd like to say is if anyone really wants to get into the philosophy of cloning, they should read a book by a South Australian novelist, Peter Goldsworthy. It's called Honk If You Are Jesus.
Well, I have to say I was going to raise that with Jenny, but she kind of killed it with the whole idea of Peter Goldsworthy's book is that they have a relic from the original crucifix and working, I think it's at Bond University, but don't tell anyone, working on cloning the man himself from it. But she really put the kibosh on it with the DNA only. But it is an interesting philosophical meandering. No more comments, and in fact, no more questions. But thank you. That has been a terrific array of questions, and we really hope you've enjoyed it as much as each of us have up here this evening. Would you join me in thanking Professor Jenny Graves, Matt Fisher, and Professor Peter Chong, all of whom have so very generously given their time. And just before we wrap up, don't forget we will hear tonight's episode on RN's big ideas in the coming weeks, and you can always podcast that or listen to the podcast for this and the earlier events, death, sex, murder and life. Also, they're all being put on the Academy's YouTube channel. Just follow the Australian Academy of Science on Facebook for updates, and don't forget Australia's Science Channel, RIOS's own channel, will also broadcast tonight's show. Look, thanks again. It's just been a great night. You've been a terrific audience. They've been a great panel. And thanks again to RIOS for hosting us. You've been wonderful. To Nova for being education department partners, and for Science Technology Australia, our series production partner. And because it is the last one, I do have to give some special mentions of thank you to the staff who've worked hard behind the scenes to make the whole series happen. Or some of them are not here tonight. Mitchell Pearcey, Ray Kellett, who is here this evening, Dion Pretorius, and also here this evening, but no longer with the Academy, Kylie Walker, whose brainchild the whole thing was. It started with an idea for a live autopsy in Hobart. It's changed a bit, but it's been a wonderful journey, and thank you very much, Kylie. And of course, again, thank you to our presenters and to the Academy. It's been an absolute pleasure. Thank you.